Yeah, well, I think we can start. So thank you very much, everyone, joining today our High-Affinity Talks. The topic today is end-to-end antibody discovery, is displays, automation, AI, and multispecifics. I am happy to welcome Andre Teixeira. This is like a great conversation to be having now. So, Andre is Senior Director of Antibody Platform at the Institute of Protein Innovation, which is a non-profit organization. And I'm Stan. I will be a host today. I'm a co-founder of MyLabs. Before we go into the actual webinar, just a few words about our company and what we're doing. We are the creators of MyTR. That's a tool that many people know and use from academia for TCR and BCR profiling. We work a lot with academia in the industry on the antibody discovery side. Today we're building and we're doing an end-to-end antibody discovery platform on the software side, on the data analysis side, and we work with many people in academia and industry who are involved in antibody discovery. That's why it's so interesting for the audience that we have, and to have these conversations with Andre today, who will be showing how antibody discovery can be put at scale. And yeah, let's go from there. So we will have first slides from Andre, and then we'll have a Q&A session. You can see in the Zoom, there is a chat and there is a Q&A section of the Zoom interface where you can put your questions anytime. And then after the presentation from Andre, we will go through the Q&A session, and feel free to ask questions. I think the topic is very hot and interesting, so we expect a lot of questions. Andre, please.
Okay, thanks, Stan, for the introduction. Oops, I need to share my screen before I project. Sure. Do you see the correct screen? Okay.
Yes, all is good. Yes.
Later. All right, so it's a pleasure to be here talking to you about the work we do at IPI. We have a very talented team of scientists here doing all this work. Bear in mind that everything I'm showing you, none of this is actually my work. That's the work of the IPI team, and just the mouthpiece here transmitting it to you. So let me start by stating the problem that we believe exists and that we're trying to solve. Oops, right. Now, if we look at the landscape of monoclonal antibody therapeutic development against the human proteins, we see that we have almost 700 clinically active programs. Those are against human targets, either approved or in clinical trials. Out of these almost 700, about 250 unique targets involved. That sounds like a lot, but it's actually very concentrated. If you look at just PD-1 and PD-L1 programs, which I'm highlighting here in green, that's a single biological system, ligand and receptor. We have 74 individual assets in clinical development or approved. That's about 11% of the entire world's pipeline. If we then look in the context of the human proteome, the extracellular accessible human proteome, we have more than 5,000 human proteins that are either secreted or in the cell surface. That can be compounded by splice variants that we're not even counting here, plus combinatorial targeting. If you think about bispecific antibodies, it's a growing field. Now you could combine any two molecules to create a new mechanism of action, which I'm listing here. Like the fact that you have antibodies against the target doesn't mean that you have exhausted all the mechanisms you can explore against that target. And now if we take all those clinical programs and we put that into the context of the classes of proteins that we're talking here that are accessible in the human proteome, you can see that's actually a very small share of those proteins that are being explored clinically. Of note here is the multipass membrane proteins, which GPCRs, ion channels, solid carriers, they're extremely underexplored by monoclonal antibodies, especially because of the difficulty in exploring them. Now, this is therapeutics, but every therapeutics born before it can become a validated target, we need to do biology, right? So if we look at reagent antibodies against the same set of proteins, the extracellular proteome, we can split in cell surface and secreted, and most of those are strongly associated with some sort of disease in different areas like oncology, neurodegeneration, cardiometabolic diseases. It's training right now, immune, etc. However, out of these heavily implicated in disease proteins, just a minority of those has publication-cited antibodies. And even if you look at the citations, most of those antibodies are for Western blot and not necessarily for flow cytometry experiments or immunofluorescence experiment. So we're lacking real tools, breadth of tools to do more biology, more discovery, so we can actually expand our knowledge of the systems that are involved in human health and can unlock new therapeutic opportunities. It's exactly what I'm describing in my next slide. So, and that's the role of IPI. So IPI has started in 2017, founded by Tim Springer and Andrew Cruz. We're a nonprofit research institute. We like to describe ourselves as a hybrid between academia and industry. And the reason for that is because we are a nonprofit academic institution. So we have freedom to pursue underexplored biology without having to be constrained by developing the next clinical pipeline or things that will generate some immediate return. We can also do open science, which is we can publish the work we do, we can share the molecules we develop, so the community can engage, test, and discover new things. However, the way we're organized, it's more similar to an industry operation in which we're building high throughput platforms. We have more discipline around our processes and our workflows. We have a team of professionals working as opposed to the common academic structure, or have postdocs and grad students and PIs, or we have specialized teams working in each area. And that allows us to operate at scale. So when we combine both, we have this, we have IPI basically doing the work I'm going to show you. So our main activity today is what we call the antibody platform pipeline, which I'm showing here at a very high level. We do everything end-to-end, from antigen production to discovery, NGS analysis of the outputs, production of the antibodies, and then characterization. And what I'm going to do today, I'm going to go into each one of these steps and tell you how we do each one of these processes. And please keep in mind that I'm showing you a single slide of each one of these processes, but each one of these could easily be entire webinar and each one of those topics. So it will be a little bit of an overview. I hope it's helpful. If you came here waiting for some heavy biology, that will guarantee that won't be the case. It will be a whole bunch of workflows and trying to share some of our expertise and what we found that works and what doesn't. And the proof, of course, is in the pudding. Here are pipeline in numbers. So you can compare calendar years 2022 all the way to 2026. So it's the current year. Of course, the year has not ended yet. So in blue, you are seeing the number of antibody binders we have confirmed. So in 2025, for example, we confirmed more than 3,000 antibodies against its intended target. Those are spread into 211 different genes. But that doesn't tell the whole story. Sometimes we take genes and we split them apart into different pieces because some proteins are big and they have different domains that have a different biological function. So if the true number of successful discoveries, it's in green, right? So in 2025, we have developed antibody binders against 258 unique proteins, which are different pieces sometimes of the same gene intended for a different purpose. And this year, although until April 28th, we have reached 133 unique targets. Everything starts with antigen engineering, right? So we don't work usually, let's say, with PD-1, PD-L1, or the popular proteins. We're looking for proteins that are largely underserved. So we have to do everything from beginning to the end. So we start with the antigen. And of course, everything starts in the construct design. This is one of the hardest parts, the parts that it takes a lot of time to do a proper construct design because you have to integrate a lot of information. We're very careful looking at structures, looking at a sequence identity to identify cross-reactivities, post-translational modifications, so on and so forth. And one thing that we have developed in-house that helped us immensely, that I'd say it's probably 10 times to 100 times faster now to develop constructs. We developed a platform that we call Open Antigens that we started using about a month or so ago. That makes us a lot more efficient, and we will release that soon to the entire community so you can use the same tools we use to develop our targets. After we develop the constructs, we go into a small-scale screening phase. Whatever passes small-scale screening is scaled up and then stored and documented for later use. And currently, we're doing more than 600 large-scale batches of antigen per year. And it's worth mentioning that we use mammalian cells for everything. Now we start with the small-scale screening after we have designed the constructs. And here I'm showing you a screenshot of our Open Antigens database. And you can where we integrate all the information we need. And we have pre-computed antigens so we can develop that quickly. We order synthetic genes, and those come in 96-well plates. All right, here's our first automated step in which those genes are automatically transfected using a Biomac i7, and then they're grown in 96-well plates, deep well plates, shaking, usually 5 to 7 days in XP293 cells. After that, we purify all those antigens in small scale using, again, a liquid handler. Those are magnetic beads, purifications, usually nickel beads. And after that, we do our first QC step in which we measure the concentration by absorbance at 280. And we recently, about a year or so ago, implemented a step doing capillary electrophoresis. And the reason for that is that we used to do an ELISA at this step. So we used to not purify, just take the supernatant and do an ELISA. And that can work sometimes, but the problem is sometimes you have secretion, but the antigen is aggregated, for example. And you do the ELISA, you get a strong positive signal. But when you try to purify, you don't get anything because what we speculate is that the antigen is not in a purifiable format, perhaps because it's aggregated. And so we found out that we have a much better success rate when we scale these things up, when we do this kind of analysis, as opposed to just doing an ELISA measurement. Right? And that's where we make our first decision. We bank all the results in our system, and then we decide what gets scaled up and what it's not working. And if we were to do all these tests in large scale, of course, that would be absolutely impossible. We need to screen everything at small scale first. So next, we scale up our targets. So we have a team of researchers that do that. So each researcher handles 6 batches of large-scale antigens per week, right? So it's not like we're trying to study a protein for a year to figure out how it works. And no, it's each researcher has 6 different proteins every week. And that's why things have to be pre-screened, and we have to be almost sure that they're going to work. Usually 200 to 400 mils of culture is enough to get the yields, but we can also get an estimate from the previous step. We aim to get between 4 nanomoles to 10 nanomoles of protein for our steps. Whenever someone asks you how much protein they need and someone gives an answer in milligrams, that's probably inaccurate because we work with proteins sometimes from 10 kilodaltons to 200 kilodaltons. That's a 20x difference in the amount you need when you're thinking in molar terms. So it's better to record everything in molar terms when you're dealing with this heterogeneity in your process. After production, we tend and affinity purify everything. Some proteins have very high expression. It would be enough to do nickel purification, but some proteins don't. If we just did nickel purification, we get a smear of contaminants. And what we found is that doing tandem affinity purification, we can get very pure protein regardless of the protein. Since we don't want to figure out which ones we need to do that and which ones we don't, we just do tandem affinity purification for everything. Worst case scenario, we get better protein than we had before, right? After we do this purification, and it's important for you to have very pure protein, especially if you're chemically bioaccumulating your molecules, because you don't want to do antibody discovery against contaminants. You want to do antibody discovery against your target of interest. After this affinity step, we do SDS page. Then it goes on to confirm that the proteins are there. Then we do a size exclusion, polishing. Everything gets QC'd. We do gel shift assays to confirm that they're labeled, and DLS to confirm that they're not aggregated. Everything gets stored in matrix tubes and registered in the system. And this way, it's easy for us to track the inventory. Important thing, we have developed co-expression of chaperones and biotin ligase in-house, so that ensures proper folding of these proteins and reduces the need for biotin these proteins downstream. Then we move into our discovery phase, right? Here, each researcher is handling 12 parallel campaigns. So think about that in contrast. Usually, a researcher is doing discovery against a single target or two targets. Here, each researcher is handling 12 different antigens. They can be related or they can be completely unrelated. But since we're using matrix tubes, 96-well formats, 24-well plates, everything kind of feeds together in a nice way. Again, we use biotin-related antigens for everything. We have two in-house libraries that we have developed, either a human Fab library or a human VHH library. I know human VHHs don't exist, but we humanized VHH scaffolds, which are about 90% identical to human, so that's why I like to call them human. And we follow a traditional selection funnel, right? We start with the libraries, we do magnetic purification, and then we after 3 rounds of magnetic purification, we do flow cytometry. And from the flow cytometry, we NGS everything. Very important for our process, you do need to do stringent negative selections. So we do negative selections during max, we do negative selections during fax, and that guarantees that we're developing antibodies against the target of interest and not just antibodies that bind something, right? It's a very common mistake in discovery campaigns to do discovery against the target and to not deplete your libraries or not even test. For first reason, you're gonna get binders against close homologs. Every protein usually has homologs in evolution, gene duplication, so you have similar proteins in the human proteome. So you always want to be aware of that, and you want to deplete against those if you want selective antibodies. We use lots of fusion tags, so we need to deplete against the fusion tags because we don't want tag binders. And of course, polyreactivity reagents, because we don't want sticky antibodies, usually heavily hydrophobic or charged antibodies. So we also want to deplete for those. As I mentioned, we have 2 libraries, one Fab library. They're both more than 10 to the 10 transformants. They're both in our Yeast display platform. The Fab library has diversity only in CDR3, heavy CDR3 and we have different germline scaffolds. So the light chains are completely constant. We just have 4 different light chains. The reason for that is when we're doing downstream validation, when I'm reformatting the clones, I don't need to worry about the light chain because I have this 4 light chains in stock. I never need to order synthetic genes for the light chains because they're all the same. And I don't need to reformat to worry about reformatting light chains, which basically cuts my cost and complexity in half of doing downstream production of these antibodies. And we don't really see any downside of having these constant light chains. And we have 4 different heavy chains. Since we barcode this heavy chain, so we know what light chain is paired with which. So just by doing a simple Illumina sequencing, we know the identity of the heavy chain and the light chain. And we also have the human VHH library, so that's about 90% identical to human antibodies. We have heavily engineered that for stability. So we want these VHHs who are prone to aggregation to be stable. And in that case, we have CDR2 and CDR3 diversity. Very important to notice, all sequence liabilities are removed from these libraries from all CDRs, including CDRH3, to maximize the success rate in the campaigns. Fact sorting gets a little bit trickier, right? So we usually start our fact sorting with actually max depletions. So we take the samples, we do max depletions against the off targets, you take the flow through, and then you do stainings in 96-well plates. When you do the stainings against the target, we titrate the concentrations. So we stain different concentrations, including 0 nanomolar, and we stain against off targets. Everything is analyzed in a high throughput flow cytometer, so we can visualize the results for the entire plate. We look at those results and we decide what's worth sorting and what's not worth sorting. So for example, I can look at the sample, I see it has great binding at 20 nanomolar, so I go and sort that sample for a different target. I can see that the binding is good at 100 nanomolar, so I'll sort that one. And that's a lot more efficient than manually running entire 96-well plate into a full-size flow cytometer, right? So we just run the samples of interest in the full-size flow cytometer. After sorting, we grow everything in 24-well deep well plates, and then we cycle that 3 to 4 times. Very important here is the affinity ramp, right? So we want to increase the stringency as we progress. So usually we never go lower than 100 nanomolar. If we don't see any binding at 100 nanomolar, we just consider that a failure because we think that there's not much use for the antibodies if their affinity is worse than 100 nanomolar. So usually we start at 120 and we try to titrate it down to 1 nanomolar. Obviously, it's not always possible, especially depending on the depletions. If you have off targets that are very close, it's harder to get very high affinity binders. We imagine if you're going to do that many discovery campaigns, that it's absolutely impossible to do colony picking, right? Because if you were to pick 3, 4 plates of each discovery campaign and then send or sequence everything, it would be very labor intensive and very expensive. So we decided to not do that, and we just do next generation sequencing of everything. So, and that's a lot more scalable than doing colony picking. So we do, and it gives us more information, right? So we sequence usually at least 4 selection rounds per discovery campaign, because you can load as many samples, not as many samples as possible, but if you have 20 million reads and we need about 100,000 reads per sample, you can say that you can load lots of samples per MySeq run. And we can also sequence if we see some off-target binding, but albeit less intense, we can also sequence that to exclude the antibodies that may have any off-target binding. And then we can do our analysis for enrichment. So we track if clones are reaching round over round, if they're depleting, and also absolute frequencies and clustering sequences and so on and so forth. One thing that we have implemented for the past year and a half, I would say that has been very impactful, is what we're calling the Delphi AI analysis. So, and that's where I think AI has been the most impactful for us in our process. So we developed this package called Delphi, which is interpretable AI machine learning to predict the properties of our antibodies. And we built it in a way that it can be extended basically for any property that you have data on and then you have an interest. So for example, if you have labeled data sets and we have lots of data on polyreactivity and antibody aggregation, we can feed those data sets to our software, and it will embed in several different ways. So you can use language models, we can use all sorts of different features. It will train multiple architectures to try to predict your data. So random forest, XGBoost, convolutional neural networks, transformers, and then we'll optimize automatically for you. So each data set and each architecture has different things that need to be optimized. So the software already tries to optimize everything for you, so you don't have to know how deep your transformer has to be. So it just finds the best architecture, and it does the cross-validation. From that, we can now predict properties of antibodies. And one cool feature that we also implemented is that we made them the model interpretable, right? So if you look here on the right, I have a panel that shows HCDR3 position for the amino acid and the likelihood of aggregation, and that's in our data. So we can run these AI models in our entire NGS sets and predict which antibodies will aggregate, which antibodies will be polyreactive, and we can also know why they're going to aggregate. For example, it's going to tell me that there is a tryptophan in position 5 of the HCDR3 that's likely to drive aggregation. And that's very important. You're going to see that we don't test many antibodies. So every antibody matters. We don't want to test things that will fail. And we were finishing to write the paper for this, and we'll publish the preprint, the code, and the models, hopefully in July of 2026. Everything moves then into antibody production, right? So we reformat, express, purify, and QC. Usually we do 24 leads per target. Like I said, if you're in biopharma, biotech, or doing drug discovery, you can do up to some people will do 2,000, 3,000 clones for a single discovery campaign. We don't, because we can't, right? So it would, if we're doing 300 targets, 200 targets a year, you need to prioritize. And that's why the NGS analysis, being very stringent on the discovery with the off-target deselection, polyreactivity, de-risking the libraries, so the libraries are optimized for good antibodies, and having good antigens, everything will end up paying off at the end, right? If your process is not optimal, you probably need to screen 100, 200, 1,000 molecules to find the molecule you're looking for. If you build a good process, you don't need to screen that much. You can drive your process to deliver what you're looking for. So 24 leads per target. We reformat IgG, Fab, VHHFC, or VHH, depending on the project. The vast majority will be IgG or VHHFC. That's our standard pipeline. Everything gets synthesized and delivered in 96-well plates of DNA. Here, I think it's the most essential step in automation. You could not even try to do this without automation. Matching heavy chains and light chains manually, if you're transfecting 300, 400 antibodies, it's not realistic. You need to combine the correct plasmids in here to do the correct transfection in the correct well. So you absolutely need an automated system to do that successfully. Or, I don't know, you have better skills than I do. Next, those are expressed in XP293 cells, 5 to 7 days. Then we go back to our automated liquid handler, do positive pressure purification, usually with protein A. We used to use magnetic beads. We moved away from the mag beads because we found that you don't really get good elution when you're working with some germlines and with VHH. So VH3, 23 germlines or VHHs, they tend to bind to protein A. So protein A doesn't bind only to the FC, it also binds to the variable region in some cases. And it's hard to elute the molecules from magnetic beads, and it works a lot better using columns and positive pressure. And then all the molecules then go to our QC step. Everything is tested in ELISA for polyreactivity, and in the HPLC for aggregation, we do size exclusion chromatography. Very important, although we do this QC, it's not a gating step for the next phase. I'm going to show you what we do next, but, and actually I can just skip to the next slide. you would think that something fails the aggregation or fails the polyreactivity, we should not test it. However, everything is in such a nice 96-well format. It would be harder to cherry pick the clones than to just test everything in the workflow. So even if the molecules fail, they still get tested downstream for binding, flow cytometry, or imaging, right? For this specific reason. So in all these steps, the sample prep is automated as well. So you take a purified antibody. It can go in 3 different tracks in parallel. First one is high throughput SPR, where we do antigen titration. We usually use 2 to 3 densities of antibody in the surface, 4 to 5 different concentrations of antigens with 4-fold dilutions between 3 to 4-fold dilutions between these concentrations. That's enough to get accurate readings for the kinetics of this antibody. We also do high throughput flow cytometry. We use CHO cells transfected with the human and the mouse version of the same gene, 14-point titration curves in 384 well plates, and then we calculate the EC50 so we can see binding against the cells expressing the receptor and not just binding against recombinant protein. And the third step, which is still under implementation, we don't yet test that for every single antibody, but that's the plan in the near future, is doing high throughput confocal microscopy for everything in 3 concentrations, so we can actually visualize the antibody binding to the cells to see if it's binding to the expected signal. Here's an example of how it looks like. So for example, for one antibody, IPI mouse neuroligand 2.4, that's the kind of results we get. So on the left, you see SPR binding. So we have the titration. Here we have 4 different antigen concentrations, and that gives us the affinities. In the middle, we have the EC50 calculated by flow cytometry in the cells expressing the target. And you can see 2 different curves, one in red against the mouse target, in blue against the human target. So these antibodies specifically cross-react between species. And we have all our controls down here, so we don't see any bindings to our control cells. And then microscopy. Of course, I wanted to show you a very nice picture. So I'm cheating here. These are not the CHO cells that we usually use for microscopy. Those are actually cancer cells that naturally express this target. But just so you get an idea that we do use confocal microscopy to validate these targets. Usually, when we get this kind of result, are not in the high throughput stage. It's downstream when we're doing a more in-depth validation of the antibodies. And obviously, I need to show you proof of some results we get. So this is one of my favorite campaigns. We usually work in entire families of proteins. This is the semaphorin family. Semaphorin are cell surface receptors. Some of them are also secreted, and they are involved in axon guidance. They're involved in immune response, immune synapse, generally involved in cell-cell communication. It's 20 different members. You can see SPR results for all 20 different members here. In this case, we have about 500 antibodies in here. 165 of those antibodies have a KD that's less than 10 nanomolar. And on the right, you see the same antibodies tested by flow cytometry against the cells. And you can see the EC50s are plotted. And we have 280 antibodies with a EC50 that's less than 10 nanomolar. Ooh, this is my last part that I'm going to show you. Also, we are doing it wouldn't be fun if we're not doing some other interesting things as well, right? So one of the things I'm particularly very interested in is multispecific antibodies. And there's one specific class that's called biparatopic antibodies. So those are antibodies that bind to non-overlapping. They bind to the same target, but in non-overlapping epitopes, right? So there are multispecific that binds twice to the same target. One nice feature is that you can get very strong cooperative binding. So if you look here, I have binder A and binder B. Those are just regular monoparatopic molecules. They have very poor kinetics, right? When I combine them into a biparatopic molecule, you can see that the combination of these 2 different binding domains gives you the cooperative binding that's much stronger than any of them by themselves. So it has this synergistic effect. Another nice thing about biparatopics is one, you can drive receptor clustering. So you can see here in the illustration that since they bind to non-overlapping epitopes, they can bind in trans and generate this daisy chain of receptors, right? If you look at the cells represented here in this microscopy imaging, you can see the cluster of receptors on the cell surface. And for a large number of receptors, this can drive receptor activation. That's true for most immune receptors, tyrosine kinases, and many others. But you can also do different things. You can lock receptors for antagonists. So for example, you can make a binder that binds to the tip of the receptor and then the base of the receptor. You have those sausage link-like receptors with Ig domains. Then you can basically lock the receptor in an inactive conformation. And this is my last slide, which I'm showing you some example of these two different behaviors. We made biparatopics against a neuronal target. So there's a cell surface receptor in neurons that can drive axon growth, right? In this specific experiment, we're taking spinal cord explants from rats, right? Those spinal cord explants, it's a bundle of neurons, basically. You can take that and do treatments ex vivo, right? So ex vivo, you can see here in this picture is a control, negative control. You see this bundle. There's not a lot of growth outside of this explant. If we treat that with the natural ligand of this specific receptor we're working on, you can see that we have intense growth of axons outside of the explant. And if we treat with biparatopic antibodies of two different kinds, for example, you can see here, if we treat with molecule 265 alone, we can get the same behavior we see with the natural ligand, which means that this antibody has an agonistic effect on this receptor. But if we treat with a different designed biparatopic, and in this case, we're treating both with the natural ligand and the antibody, right? So the natural ligand is supposed to drive the growth, but when we treat with both at the same time, we see that the antibody is able to prevent the effect of the natural ligand. So you also develop an antagonist. So it's an exciting type of antibody that's, I believe it's underexplored. Most people are working in T-cell engagers when they think about multispecifics. But that's one of the fun things we're doing in this area. So lastly, like I said, I need to acknowledge everyone who's doing all this work, right? So I want to thank the antibody platform teams who do all the work from the antigen and antibody discovery. We are, I like to say, we're the blue-collar scientists. So we do hammer out a lot of molecules, it's a lot of production, it's a lot of grind. Downstream of our teams, we have a very talented team of scientists in our characterization group, automation and lab operations and neuroscience, who do a lot of cool science characterizing these molecules and automating our process and exploring implications in neuroscience. Also, thanks to our collaborators and founder. And with that, I'm happy to take any questions.
Yeah, well, thank you very much, Andrea. That's exciting talk. This is super interesting, and we'll really have a lot of questions. But before we jump into all the questions, I have three from myself that I want to ask. I don't want to eat all of them. The first one, which is really interesting to me, you said that the colony picking is not viable anymore. And in your opinion, is that universally true for any campaign, or it's just for you because of the scale and throughput that you're operating in?
I don't think colony picking will ever match the amount of data in NGS. I think the only advantage of colony picking is just faster validation. So you can just pick the colony and test binding right away, right? So you bypass the NGS, and then you bypass the synthesis and the production, and then you can get like a faster turnaround. If you do the math, but now you have to deal with all this. If we're talking about $9 per sample just for Sanger sequencing, and you're picking 4, 5, 6, 7 plates, you do the math, you're going to spend $10,000 just in Sanger sequencing to identify for a single campaign, plus the work of picking all these colonies and processing. And it's really, I don't think nowadays is the best way to go about these things. Gene synthesis is so cheap.
Interesting. But in terms of the quality of the leads that you can get from NGS and colony picking? In your experience, like, can you identify better leads with NGS?
I think so. We know, I mean, in the display field, I'll talk about display specifically. We know historically that frequency does not necessarily correlate with affinity, right? So when you were doing colony picking, you're restricted to your most frequent clones, right? You can't dig deeper. You can't have a cluster. You can't have a bird's eye view of your entire population. You can't track enrichment. So I think you can get better results, especially if you're looking for properties like what enriches when you decrease the concentration, what enriches when you switch the target, so you can have a much better decision making.
In this regard, go into the computational side in your Delphi algorithm. The question that I have, it's so we're looking forward to see the preprint, all of us, and the AI and machine learning approach that you developed, is it specific to your platform, to how you design your synthetic libraries? You mentioned that you do diversity in CDR3, for example, not in other CDRs, or it will be more like a universal framework that can be used in other campaigns and other library designs?
It is universal. So specifically, it depends on the architecture. So it will accept any VHVL. You put it in there. It can be even missing the VL, right? Because if you're doing VHH, and it will be trained on the entire sequence of your antibody. In some cases, for example, when you're training a transformer, you have different architectures where you can add attention pads. So we put on a special attention on the HCD3 because we know beforehand that you get better results if you pay attention to the CBA3. But it is universal. You can train any data in there. And you can extend our training data as well. So we'll publish the weights. The weights will be public. So you can start from our checkpoints and you can add your own data.
Cool. That's we're looking forward to it, really. And the last question from myself, just to fulfill my curiosity. So in your biparatopic antibodies, so when you pair them, do you always see this increase in the affinity, or it's, you need to tweak, play around, and find which is the pair that will work actually?
Oh, you mean a VHVL pairs that will work, or?
No. The your biparatopics, when you take two
We have
binders.
our own yes, no, we have our own secret sauce. I cannot reveal what there are ways to do it. I would call it, there are naive ways in which you can just muscle through and test a whole bunch of things and see which ones work. It's not as trivial as you just combine two binders. We have developed our own methods in-house in which it's basically one shot, one kill. We know which binders we can combine to give us the cooperative binding. Unfortunately, I cannot tell you today how exactly the sausage is made, but it's there is some finesse in there.
I see. Interesting. Okay, now I'm switching to the questions that we have from the audience. We have a lot of questions. So the first one is, do you do transmembrane receptor antigens too, like nanodisk formats?
Yes, we have started implementing this about a year ago. We started using, we're building different platforms. So we have detergent solubilized molecules. We are using peptide disks as opposed to nanodisks, just because they're slightly faster and easy to work with. And we are betting a lot on VLPs, virus-like particles, where you transfect the antigen with HIV gag, you produce VLPs, then you can do discovery against the native form. Obviously, it's not nearly as mature as our soluble pipeline that I showed you with the SEC, tandem purification. And we have had success with a few targets, but it's definitely not, we cannot do 300 of those a year. So it's a lot more intricate at the moment. But I'll say that my commitment is that we'll industrialize that as well. So that's our goal.
Thank you very much. Next question is, what is your typical affinity coming out of Yeast display from synthetic libraries?
Like I showed in that, it's highly variable. Like I showed you the semaphore example. Basically, a third of the antibodies have less than 10 nanomolar affinity. It's very target dependent. If you have targets that are LRR domains, beta propeller domains, they're very rigid. You tend to get really good antibodies. You can get several single digit sub-nanomolar antibodies. If you're working with unstructured proteins, very difficult to get good antibodies because it's just how it works. And it depends on if you have a lot of, you're selecting against target A and deselecting against target B. Target B is 90% identical to target A. It's harder to get higher affinity antibodies as well. But I would say, as a rule of thumb, I'd say 1/3 of the antibodies should be less than 10 nanomolar affinity.
Thank you. The next question, how are the human VHH humanized?
Oh, yes. Well, I would say, I'll give you the general, I won't tell you exactly which mutations there are, but I'll tell you in general terms. So the first thing we do, we start with VH323, which is the closest homolog to a VHH, right? And then you do what everybody does, you do the 4 mutations in framework 2 that will stabilize the framework 2 and abolish VL binding. However, that is not enough to stabilize a VHH, like doing these 4 mutations. So we did an extensive campaign of express, let's say 200, 300 molecules to figure out variations that would give us stable humanized VHHs. So we ended up using 2 different scaffolds. So our VHH library has 2 different scaffolds with slightly different mutations. And like I said, they are about 90% identical to human VH323. But it's challenging. It's challenging. One would be led to believe, if you read the literature, every introduction of VHH paper will tell you that VHHs are incredibly stable. They're wonderful. I have never heard this of anyone who actually works with VHH and do size exclusion HPLC testing. VHHs are much harder to work with than regular antibodies. They tend to aggregate a lot more. If you're doing extensive HPLC analysis, you will observe that.
Thank you very much. The next question is, how do you multiplex sorting? And how long does one round of sorting take?
Yeah, so the multiplex, the sorting, we do the stainings in 96 well plates, right? So think we have 12 antigens. So each column is an antigen, right? Each row is a different condition for that antigen. So if you think about the easiest way to design your plates, let's assume, for example, you have highest concentration to lowest concentration. That's like 4 samples, 5 samples, ending with a 0. Then you have off target 1, off target 2, off target 3. Everything can be done using multi-channel pipettes. We have an attune flow cytometer, right? That would put that sample then into the attune flow cytometer. You have templates that will analyze the data for you, and then you see what samples look good. So now you basically have 12 samples to sort. If you have 12 targets, you just need to select which ones are the good ones. You put in your tube, and then you do your sorting. It's a whole day. It's a long day of work, but you can do the 12 sortings in a day if you are, not everyone probably, but our team is extremely talented, so they can do 12 sorts in a day, our discovery team. You should ask them, not me.
The next question, so we have, all questions are very interesting. The next question is, hi Andre, how many were you initially producing and screening for to generate the data that then gave you the confidence in the 24? That you're selecting now 24, but initially, did you start with hundreds and then optimize?
Not really. Like the reality is you only have so much money and so much bandwidth, right? So it doesn't matter how successful you are. I only have the bandwidth to order and test 24. So they need to work. So what happened in the past is that we would try molecules and it would have less success. Then it would get 1 binder or 0 binders. And as we get better, we get 10 binders, 15 binders, 20 binders, or 24 binders, right? I wish I could do 1,000 molecules per but we all have to follow strict, we have to stay true to our budgets and to be efficient with our resources.
Sure. Next question. Have you thought about ways in which you can learn across multiple antigens? For example, generalizable models instead of antigen specific. What are your thoughts on this?
That's a good question. So, for example, for biophysical properties, those are antibody-specific characteristics, right? So the antigen doesn't matter. Right? We just analyze the antibody sequence. Now, if we're talking about binding, that's a different story. I don't know if I have an answer for you, right? We are engaging in some AI machine learning training. I would say the kind of data we produce is very sparse. When you produce affinity maturation data, for example, you have several variations around the same clone. It's very easy to train models there because it's just because of the knowledge density that's in there, but it doesn't extrapolate outside of your distribution, right? It only works for that specific case. In our case, we have a lot of data for different targets, but the data is very sparse. You have one clone here, one clone there. Those two clones have no relationship amongst themselves. The antigens have no relation. The families have some relationship amongst themselves. So I would say the kind of data we produce in a discovery campaign is very sparse at the moment to try to generalize across these different domains. However, you never know. The AI tomorrow, I think this is true today. I don't know if that's going to be true tomorrow or next week or next month or next year.
Well, what will be tomorrow or next year? So, things are changing
It's
very
evolving.
fast.
Yeah, it's evolving very fast.
Yeah. Next question. Are you building ways to predict epitope targeting and function in the way to how you triage antibodies for production?
Yes, and yeah. So it's computationally, I'd say that's the problem I'm the most interested right now is to predict how an antibody binds to an antigen. I think largely antibody design has not been solved yet, mainly because the previous problem has not been solved yet. We cannot predict yet how an antibody is binding to the antigen. Given an antigen sequence and an antibody sequence, how do they connect to each other? So there's a lot of effort in our computational biology team right now going into solving this specific question. It's not a solved problem yet. We're looking for ways to leverage our large data sets in a way that we can try to solve this. And I forgot what was the second question.
That's basically the question, and then you answered. So yeah. The next question is, how do you obtain reliable binding kinetics for a multimeric antigen using HT SPR, given the risk of avidity and rebinding efforts?
You don't. Whenever you have a dimer, trimer, you get what we call apparent affinity, right? You don't get reliable affinity reads. And there are two. So first of all, if the antigen is naturally a dimer, we don't really care about the monomeric affinity. If it exists in nature as a dimer, it's always going to see the dimeric form. So I think people, when they're doing discovery, if we're doing like a biology mechanism and deep dive into these molecules, it may be important for you to get the monomeric affinity. If you're developing a binder against a dimeric molecule and the molecule is always going to be dimeric, I think it becomes a unnecessary complication to try to get the real. Although you 100% you do get avidity. That's why also test against the cells. You also get the avidity effect on the cells because you have many receptors. It's going to bind multiple receptors at the same time. But in our case, I don't think that's a significant issue. With the exception, if we are not artificially dimerizing, and so a lot of people use Fc fusions to express antigens, and that's a problem. If you do an Fc fusion of an antigen that's naturally a monomer, and now you have artificially a dimeric antigen, then you have a problem because now you're measuring the wrong affinities because the antigen does not exist as a dimer in nature. So then you're just measuring the avidity.
Thank you, Andrea. We have 5 minutes and we have way more questions. So I will try to let's try. So for constructing large and diverse yeast display libraries, how do you compare yeast mating versus direct cloning approaches? Which method typically gives the largest effective library size and better diversity?
If you think about in theory, theoretical principle, mating obviously can give you a lot bigger libraries, right? You can because let's say you can transform a 10 to the 10 library. If I can transform two 10 to the 10 libraries and I mate them, in principle, you could get a 10 to the 20 library. So theoretically, mating would be superior. However, there's a catch. Then that's something, like I said, every slide in the presentation could be a whole presentation in itself. If you do the math, 10 to the 10 library, for each antigen, you need to cover diversity at least 10-fold to 100-fold diversity in your round one. Now you are growing yeast, and then we usually grow 20 liters of yeast overnight in the library to do the first round of selection because we have a 10 to the 10 library. If you have a 10 to the 11 library, and then you have to have good coverage, you have to 10x your growth, you would be growing 200 liters of yeast. Now, if you have a 10 to the 12 library, you would be growing 2,000. I don't think it's, people advertise, oh, I have a library that's this big, that big, that big. No one is growing 200, 500 liters of yeast. So you're only sampling a subset of this library. So realistically, I'd say you can just transform, get your 10 to the 10 library. It will probably be good enough. It's unlikely that you'll be able to sample more than that.
Well, thank you very much. That's very, very interesting answer. Next question, is colony picking still not viable if you're looking for specific functional properties, specific agonists? How might NGS enrichment be used to detect specific functional classes?
Yeah, so it's a very good question. I'd say NGS, in this specific case, I'd say NGS is far superior, right? If you, for example, we have an agonist project in which we have designed the receptor in two different conformations. We have an inactive conformation and we have a locked active conformation. And now we do discovery against the active conformation, depletion against the inactive conformation, and then we can enrich for binders that differentiate. So, and the rationale is, and usually that's true for GPCR, that's true for several classes of receptors. Whenever we stabilize the active conformation, you drive agonism. So antibody finds the receptor, it changes the receptor into the active conformation and activates the receptor. Some receptors are harder, some receptors are easier. The harder it is, the more rare your clone is going to be. The more rare your clone is going to be, the harder it is to find by picking colonies, right? So the trick is design the discovery well, design the antigen well first. Then you design the discovery well, and I would say then you design your screening or NGS in a way that you can detect those. So I'd say you're right, like functional binders, it's a lot harder to develop. The more information, the more you build into your process, both in the NGS analysis and the upstream processes, the more likely you are to recover that rare clone that has the function you want.
Thank you, Andrea. The last question. So are you going to publish ever the NGS raw data counts and enrichment from your campaigns that you regular?
We did already actually for 12 targets. There is a bioRxiv paper, I think it has just been accepted at Cell Systems, in which we published extensive data sets, both of naive library and discovery campaigns and enrichments for 12 different targets. We will likely publish more. Of course, it's a lot of work and time, but we plan to, but we'll see how that goes.
Well, thank you very much, Andrea. That's super nice conversation. A lot of questions we didn't answer. We don't have time, but thank you everyone who attended, who asked questions. We will send you, Andrea, the list of questions, and maybe this will be helpful for you. That because we have a lot of it. Thank you very much, all, and we will make